complex to see that today, but so glucose is transformed into pyruvate. If you have anaerobic conditions, you produce lactate. And after that, pyruvate enters the Krebs cycle in mitochondria. And with the production of ATP, NADH, FADH, etc., for further oxidative phosphorylation. And at the end, oxidative phosphorylation occurs in internal membrane of the mitochondria. And at the end, ATP is produced, and this is the energy of cells. So this is completely dynamic. So metabolism is dynamic. Metabolomics is static. It gives a snapshot of the physiology of the cell at a given moment. So we say that it provides a unique fingerprint that specific cellular processes leave behind. This is important. This is a fingerprint. You have questions? So, why is it interesting to do metabolomics? As I said you before, because metabolites reflect what has really happened in the cells, while DNA, RNA, and protein are not do not reflect what has really happened in the cell. So this is why people are so interested in metabolomics nowadays. So there are an increasing number of papers on scientific literature about metabolomics in different fields, in neuroscience, in cancer, in cardiovascular risk, etc. We say that there are mainly three domains in which metabolomics is used. So for a mechanistic purpose, this means that you from your fingerprints, we will extract some biological hypothesis. As I said, metabolomics is static. This is not dynamic. But if you find metabolites which are dysregulated, this give you some biological hypothesis that you will study, further study. So this is the biological part of metabolomics, but this is mainly used for classification, prediction, and prognosis to discriminate pathological from normal. So I will show you some examples. So we have the metabolic profile or the fingerprint of a pathology compared to normal state. To predict also treatment efficiency, to predict a risk, to develop a cardiovascular pathology or to relapse from cancer, for example. And this is what we call personalized medicine. And for diagnosis purpose, also to improve diagnosis. So I will show you an example from my study. So we have developed a diagnosis for Parkinson's disease, which is based on a metabolic profile of blood sample of animals on one side and patients on the other side. So we have developed this new diagnosis, which is based on the metabolome of Parkinson's patients. So this is a very important part of research now. And also there are people who can use metabolomics during a surgery to help the surgeon to resect brain tissue, for example. I think that they have an example. So this is a very important field of research. And in both parts, so in basic research, what we call basic research in biology and in medicine, or in clinical research to improve classification, prediction, and prognostic of patients, and also to find new diagnosis tool to improve diagnosis of different pathologies. So I mainly work on this part and this part, so I will show you some examples. So why metabolomics? So in 2007, so you can see this is what it is. A paper in Nature where the author, so Ellen Wilson, said, can body fluids succeed where genomics and proteomics have failed? So imagine a routine checkup, medical checkup, where your doctor take a urine sample and can report your risk of type 2 diabetes. Is normal, but there are hints that you are arteries. So a similar scenario has been provided for the past 20 years by those working in genomics and proteomics, but has not yet materialized. So 20 years ago, people were made these constatations that genomics and proteomics were failed, yes. And so metabolomics started to develop at the period, and this is also because analytical methods have largely developed and have been improved, largely improved, and also statistical tools have been developed to build this fingerprint. I will show you how it works. It's a little bit complex. This is multivariate statistics, but this is the basics of the omics, also for proteomics and genomics. So we use multivariate statistics to build what we call a metabolic profile or a metabolic fingerprint, which is characteristic of a given state, so pathological or healthy. And also, yes, this is an important paper during Nicholson from the Imperial College of London, who also launched what he called the Phenome Center. So Phenome, this is a little bit different from Metabolome. Yes, this is to examine the effect of gene, lifestyle, and environment on the metabolome. This is what they call the Phenome. So now there are several Phenome centers in the world. In France, I don't think so. So they use, they study a lot of pathology. So I will give you some, explain how it works. So this is two important points. So the previous paper from Nature and this important paper, Jeremy Nicholson, it is at the Imperial College of London, and he was a specialist in NMR at the beginning. He was a chemist, and now is completely involved in clinical application of metabolomics. Yes.
For this paper, the Phenome is to examine the effect of gene lifestyle movement on the metabolome?
Yes. Okay. Well, the Phenome is not very used. This word that yes, because we consider that the metabolome is largely influenced by the gene, lifestyle, and environment. And there are several papers which show that. This is why they opt to do what they call personalized medicine. So, also, this is this slide to show you how small molecules are important, because small 95% of diagnosis clinical assays tests for small molecules. All known drugs are small molecule, and 50% of drugs are derived from pre-existing metabolites. So you can see that, and also small molecules serve as cofactors and signaling molecules to thousands of proteins. So these small molecules are fundamental in life and in medicine and in pharmacy, pharmacology. So for this first part to convince you that small molecules are important in biology, biochemistry, and in life. And this is why people started to study the metabolome. But one difficulty is that, as I showed you before, the metabolites they have much fewer metabolites than enzymes and genes. So this is always the same molecules that you will find in all pathologies. And what is different between pathologies, between stress, this is the profile, the metabolic profile. This is a fingerprint. You know, you all have a different fingerprint, and this is the same for metabolomics. We built a fingerprint, so it's always the same molecule because the set of metabolites is small, but this is a combination of all these metabolites, which is specific of a given pathology, a given reaction, a given stress. So, an example of this is a general workflow to do metabolomics. So we start with a biological issue. We have a question, which is either mechanistic, from basic research, or which is clinical. And after that, it is necessary to design the experimental set and the sampling step. Because as I said to you before, the metabolites are very rapidly degraded. So it is necessary to choose the sample which are the most representative of what we want to study, but also which can be relatively kept during the sample step, which is not too much destroyed during the sampling step. So there are a lot of different samples which can be used, as I said to you before. All the fluids are studied, the saliva, so the very, very different fluids. The organoids, like, of course, mitochondria, embryon, etc. So this is the first point is to choose the good sample which will give you the most relevant information. After that, it is necessary to choose good analytical tool to analyze the metabolites. So there are mainly two methods which are used. This is NMR, so nuclear magnetic resonance, and mass spectrometry. Are there anybody who know the NMR or mass spectrometry here? Nobody? No? Okay. So this is methods which come from chemistry. I will give you some examples and will explain you the basics of NMR. This is an important point because NMR is non-destructive and mass spectrometry is destructive. So we will see how to choose a good analytical tool. After the analytical flow, there is a processing flow, which is also very specific. And as I told you before, we use multivariate statistics to build this kind of graph. A little bit complex, but interesting to have the basics of statistics. After that, we can use different tools to identify biomarkers, so what we call biomarker. This is the metabolites which differentiate a state from another state, or which allow a diagnosis, a prognostic, etc. And this step is also very important to validate the data that the biomarkers that we have found and to interpret them. So all the metabolic pathway, and so there are specific tools to understand in which part of this graph the dysregulation are located, and what does that mean. And after that, back to the first point, which was the biological issue. So this is a general workflow of a metabolomics study. A little bit more in detail. So you know that to design an experiment, we have to choose the good number of samples to have a statistical power. Sampling, so stop metabolism rapidly. This is a difficult point. Rapid collection of sample, freezing, extraction or not of the metabolites, storing at -80, because at a higher temperature, there are degradations. At -20 in your freezer at home, there are a lot of degradation and the enzymes work. And we can observe if we do NMR or mass spectrometry that there are a lot of degradations of the food and so on. So there are reactions are working at, enzymes are working at -20, and there are also chemical degradations. So to avoid that, we need a -80 freezer. The analysis, so NMR or mass spectrometry, but here this is NMR because I have a specialist of NMR. After that, the multivariate statistics with different algorithms which have been developed, and in MATLAB or in Python also there are a lot of tools which are available to do this kind of statistics. So if people want details, I provided some details, but it's not it's complex. I guess it is in that sense. If you do NMR during your internship, maybe you will use this kind of information.
What was the main difference between NMR and mass spectrometry?
I will explain that later to show you the basics of these two analytical methods. So, and after that, after all this, so there are a lot of algorithms which have been developed. Some of them are free. This one are free. And some of them are very expensive, genomics. C-class is expensive algorithm, but some of them are free and they are very performant. And after that, we do univariate statistics, so t-test, student test, or ANOVA. So this is classical statistics. And we can also conform the metabolomics data to other omics, proteomics and genomics. So, in this global workflow, as I told you, we select the relevant biological sample to study what we want to study. So either all this kind of sample. And after that, it's necessary to use high-resolution analytical methods. And the two most important analytical methods, high-resolution analytical methods, are NMR and mass spectrometry. So you have absolutely no notion of what is NMR and mass spectrometry? Nobody? No, there are no chemists here. So, okay. So as I told you before, metabolites are very different. There is a big diversity in structure and chemical properties. and also a large range of concentration from the micromolar to molar. And this is a complexity. So this is why there is no universal detector or analytical tool for metabolomics. So now I will give you some basics about NMR, nuclear magnetic resonance, because this is an important point. And maybe you know MRI. Maybe have you had you an exam, MRI exam, a scanner, and this is the same principle, except that for MRI, for imaging, the magnet is horizontal, while there is a big hole for you to go inside the magnet, while for spectroscopy, the magnet is vertical, and there is a very small hole to put the sample. So to do NMR, there is 3 conditions. So that's the nucleus that we want to study as a spin, a nuclear spin. So this is a quantum mechanics. So I will not develop that. What is important to know is that all there is a lot of nucleus which have no spin, but the proton H, C13 and phosphorus 31 have a spin. So they can be studied by NMR, and this is important nucleus in biology. It is necessary also to have a static magnetic field. So this is what I said before. For imaging, there is a big magnet, so you go inside the magnet. This is a static field. And when we put this nucleus into the magnetic field, they align along the magnetic field. And after that, this is the third condition, is to exit these spins to produce a resonance frequency. So 3 conditions to do NMR: to have spins, so only, for example, oxygen has no spin, so it cannot do NMR of oxygen. But there are isotopes which have spin. For example, C13, this is not the main isotope of carbon. The main isotope is C12, but C12 has no spin. So this is why we study C13, but the natural abundance is very low. This is only 4% of carbon, which are C13. The magnetic field. This is a very high magnetic field. This is 1,000 times the Earth's magnetic field. One of the biggest magnetic fields is there in Grenoble and in Lyon, but now they are biggest magnetic field. This is a challenge, a very high technical challenge. So what I said you before, oxygen. This oxygen isotope has a spin, so this is the most studied nucleus. But also all nuclei do not have the same sensitivity. This is an intrinsic property. This is quantum mechanics, so we must admit that. So the most used and the most sensitivity is for hydrogen. So hydrogen is everywhere. In all molecules there are hydrogen. So this is the most studied nucleus. And for metabolomics, this is the most studied. 99% of metabolomics study use proton NMR. So they study the H, which are in the molecules. Phosphorus is very interesting because for energy, ATP, there are phosphorus, but the sensitivity is much lower. This means that 10 minutes are necessary to analyze one sample of serum or urine using proton NMR. And if you want to use phosphorus, you see the difference in sensitivity. You need hours to record the phosphorus spectrum. And C13, you see the sensitivity is very low. So, for example, here in the EBS, so Institute of Structural Biology in Grenoble, they label the molecules with this isotope. We can, using chemistry, you can replace the isotope which has no spin with the C13 isotope which has a spin. So this is chemistry. So they label the proteins with azote 15 and C13 to do biological structural biology on proteins. So this is a way of increase the sensitivity of these nucleus. But for metabolomics, we do not label anything. We study the natural sample. And so this is why we use mainly proton NMR. A little bit complex, this one. Just the basics of NMR, very basics of NMR. As I told you before, there are some nucleus which have a spin, but this spin is like that. Without any magnetic field, the spin are not aligned. When you put your sample into the magnetic field, so the big magnet for spectroscopy or for imaging, so you have the very big magnetic field, the spin become like that. They are aligned along the magnetic field. So this is the main direction of the magnetic field. For spectroscopy, it is vertical. For imaging, it is horizontal. So when you go into the magnet, the spin of your brain, for example, when you do a brain scanner, align along the magnetic field. This is not dangerous. So you put, if you do not have this magnetic field, you cannot do NMR. The spin are like that. They are isotropically oriented into the space. With the magnetic field, they align along the magnetic field. And after that, there is a little difference, a very small difference between the up and down spins. And when we exceed them, so this is the equilibrium position. When we exceed them, they are pulled out of the equilibrium position, and we stop the excitation, and they come back to equilibrium, and this produce a small current, electric current, and we measure this current. This current contains the signal of all the protons which are into the sample, and they all have a different signal. And so this is the very, very basic of NMR, but it's interesting to know that. So the spin align along the magnetic field. This is the equilibrium position, but there is no signal. To have signal, this is like in all spectroscopy. To have a signal in spectroscopy, it's necessary to pull this is the spin out of the equilibrium position. And when we stop the excitation, so the excitation lasts some microseconds. Yes, some microseconds. And when we stop the excitation, they come back naturally to equilibrium position by doing this kind of spin, this kind of movement. And the detector is then, and the detector detects this kind of signal. This is the free induction decay. Because at the end, the signal vanishes, because there is a loss of energy into the matter. So at the end, we have this kind of signal. The signal contains the signal of all the proton into the sample. And after that, we do a Fourier transformation to transform a time domain signal into a frequency signal. And here you have a peak, different peaks at different frequencies. This is the proton, which are the hydrogen, which are in your molecule. which gives this kind of signal. And each molecule, each metabolite, has a different signature because there are different hydrogen groups. And in a serum or urine sample, there are thousands of hundreds of molecules. Each molecule has a specific signature, but you see the sum of all the contribution of all the hydrogen group of all the metabolites. So after that, we need some tools to deconvolute all this signal. So NMR is very interesting in metabolism because this is non-destructive. Of course, when you do a scanner, in an MRI scanner, you are not destroyed by the scanner. This is completely inoffensive. So this is completely non-destructive. So this is important because for traceability purpose, your sample can be restored, frozen, etc. So this is very important, this non-destructive property of NMR. This is completely quantitative. Completely quantitative because here, the area of the peak is directly connected to the concentration. So it is very easy to convert the area of the peak into concentration. The most difficult part is to identify which metabolite you have detected, but this is a very important property. NMR is completely quantitative. This is yes, very, very reproducible. So very robust. There is no batch effect. No, if you analyze a sample, a part of your sample one day and the other part another day, there is no batch effect, no experiment effect. So this is very, very important for multicentric studies in clinical research. It's necessary to analyze samples in different hospitals, in different centers. And so this is a very important quality of NMR. Very rapid, so there is no sample preparation. This means that you can take your blood sample, your urine sample, put a little bit, a small part of buffer, and you can analyze directly your sample. So in 20 minutes, you have analyzed the sample, collected the spectra, quantified the spectra. So this is an important part, an important quality of NMR. And the problems with NMR is that it's much less sensible than mass spectrometry. This is not a sensible method. So we detect micromolar, but not nanomolar. Yes, all molecules cannot be identically detected. Depends on the mobility. Because if your molecule is fixed on a protein or if macromolecules have a lower mobility than or lipids have a lower mobility than small molecules, so peaks are broadened. If you decrease the mobility, you decrease the mobility of molecules, you increase the width of the peaks, and at the end, the information is lost. So this is not a push-button analytical method. No. But now, a lot of steps of NMR have been automatized, and it is much simpler than in the past. We can also do fluxomics with NMR. It's something, so this is a dynamic part of metabolomics, but I will not develop there. So, to increase sensitivity, you can increase the acquisition time. If I acquire a spectrum of a serum sample or a cerebrospinal fluid sample during 10 minutes, and the peaks are very small. I can increase, as the sample is not destroyed, I can increase the acquisition time to 1 hour, for example, to increase the signal to noise ratio. This is the basics. If your peak are into the noise, you cannot distinguish them. If you increase the signal to noise, you increase acquisition time, the noise is the same. Of course, when you increase acquisition time, the noise is always the same because it is not coherent. Noise is not coherent, while the signal of proton increase, so you increase the signal to noise ratio. So if you increase the magnetic field intensity, you increase sensitivity, because sensitivity is directly related to magnetic field. So the most widespread B0 for NMR is now this is 11 Tesla or 14 Tesla. And they are, the highest is 28 Tesla, but the cost of the machine increase exponentially with the magnetic field. So from hundreds of thousands of euro to million and million. So this is a problem. And also there are new technical developments now to increase sensitivity of NMR, use proton polarization, but this is very complex methods. And now they are not suited for metabolomics and for clinical metabolomics. Too complex. This is a lot of very big machines. So until now, there are developments. This is physicists who developed these new tools. But until now, they are not available for routine metabolomics. So, as I told you, proton, so hydrogen high resolution NMR is the most adapted and used in metabolomics. So the sensitivity is in the micromolar range. This is quantitative, for time computing, adapted to high throughput. And each molecule has a specific spectral signature. But there is a lot of peak overlapping. As each molecule has 2 or 3 peaks, and if you have hundreds of molecules, so all the peaks are overlapped in proton NMR. And of course, water, H2O, there are 2 protons, and the concentration of the proton in water is much higher than the concentration of proton in metabolites. So, so I will show you how to. So this is the proton NMR spectrum of alanine. So you can see alanine, CH4, CHNH2, COOH. So the spectrum of alanine is like that. So there are 2 peaks, 1 doublet and 1 quadruplet. So I will not explain in detail, but if you have all these metabolites in brain, for example, the proton NMR of brain will be the sum of all these contributions. So this is why you understand that there are a lot of peaks, and it's complex to identify the individual signal of all these separate metabolites. So this is one problem of proton NMR. This is a proton NMR spectrum of brain tumor with you can see big lactate peak. And also, so this is one problem of proton NMR, the overlapping of a lot of peaks, and water. Water signal is very, very intense compared to you can see the millimolar, micromolar range of the metabolite, and this is water. So we have tools to avoid that. We can irradiate the peak of water during eclipses, a specific what we call pre-saturation of water, so to recover the signal of metabolite, and also we use deuterated water. So this is where proton have been replaced by deuteron. This is the isotope of hydrogen. This isotope has no spin, so we use deuterated solvent, deuterated water, deuterated chloroform, deuterated methanol, etc. So to suppress the signal of this solvent, because we do not detect, when you detect the proton, you do not detect the signal of deuterium. So phosphorus NMR, so interesting for energy metabolism. Phospholipids also, and C13 is has a very low sensitivity. Several hours we did for spectrum. What is interesting in C13 NMR is that the frequency range is much bigger than the frequency range of proton. So the peaks do not overlap. The broad frequency domain, no peak overlap. This is mandatory for structural elucidation of molecule to do proton and C13 NMR. And also it's very useful, there are a lot of commercial substrates where C12 has been replaced by C13. So lactate, pyruvate, a lot of substrates can be bought in a commercial society like Sigma, etc. And this is interesting because to study the flux, and this is a method to understand. So if you give to cells, for example, glucose, which is labeled with C13, so the cell will use this glucose, and so the C13 nucleus will be inserted into the product, so into pyruvate, into lactate. After the biochemical reaction, the C13 nucleus, which come from glucose, will be inserted into other molecules. And with this method, you can understand at which point of Krebs cycle, for example, there is a problem. So this is a very, very important part of research. This is the study of metabolic flux using labeled substrates. So mainly labeled substrate with C13, and also with deuterium, so H2. Deuterium, this is the isotope of hydrogen, H2. So the natural abundance is low, but it's easy to, with organic chemistry, to incorporate deuterium into a substrate. And now, at the moment, in my lab, we have an experiment with glucose labeled with deuterium and lactate labeled with C13. And we study the prematurity in babies, and we have developed an animal model of prematurity, a rat model of prematurity, and we give them lactate labeled with C13 and glucose labeled with deuterium. And 2 hours after, we collect blood and we study all the metabolites to understand what is the turnover of lactate and glucose in prematurity compared to normal states. So we have a big project with metabolomic, in vivo metabolic flux. So this is a very important point. This is not really metabolomics, but yes, this is a part of metabolomics. Also, I don't know if you know the lactate shuttle theory. This is a theory which explains the functioning of astrocytes and neurons. So with metabolic flux, people have understood that lactate is not a waste of cells. This is an energy product which is used by neurons. So it is provided by astrocytes to neurons when they need energy. So this discovery is very important in neuroscience, and it has been discovered using metabolic flux, NMR, MRI, etc. So there are a lot of applications to brain energy metabolism, but also to study cells metabolism using cultured cells. So this is an important point of research. So NMR spectrum. So when you in NMR, what is important to understand is like each nucleus, proton for C13 and phosphorus 31, have a resonance frequency, a given resonance frequency. This is like when you listen to radio. If you want to listen a given radio, you must be to the good frequency to hear this radio. Each nucleus has a given frequency. So if I perform proton NMR, I am at the frequency of proton. I do not see the other nucleus. I only see the hydrogen nucleus. This is the same for the other nucleus. So they are complementary, but it is not always possible to do all this spectroscopy on the same sample, because they degrade. For example, biological sample degrade. C13 is a very low sensitivity, so it's complex. And for high throughput metabolomics, this is mainly proton NMR. So this is proton, so hydrogen is one proton. So for metabolomics, there are several different samples which can be studied. So biofluids, so this is liquids, but also tissue, brain, liver, lung, etc. Cells, bacteria, etc. Organoids, and NMR, high resolution NMR. You can only have high resolution NMR, so this is with very sharp peaks, with liquids. So if your sample are not liquid, the spectra are like that. So cannot be used. There is no information. This is one problem also. So for semi-solid samples, and in most of metabolomics study, we must extract the metabolites from the sample. So there are a lot of extraction methods with perchloric acid, for example, or with chloroform, methanol, etc. So it's necessary to destroy the sample and to add different solvents to extract the metabolites, which are hydrophilic compounds. Metabolites are hydrophilic. If you want to analyze lipophilic compounds like small lipids, you must use three-phasic solvent. So after the extraction process, in the hydrophilic solvent, you will have the metabolites, hydrophilic metabolites, and in the lipophilic part of the mixture, you will have the lipophilic compounds. So even if NMR is not destructive, this is not a destructive method, to study a semi-solid sample, it's necessary to destroy them, to extract the metabolites, which are intracellular metabolites and so on. So there are a lot of methods which do not give the same result and the same. There is a method which is very interesting and which I use in the Grenoble Institute of Neuroscience and in CEA also. This is what we call the magic angle spinning NMR. So how to explain that? So Why, with semi-solid sample, we lost resolution? Because there are a lot of interaction between the spins. This is quantum mechanics also here. There is a lot of interaction between the spin, and this interaction led to a loss of energy in all the spin, and the loss of energy led to this kind of peak. And so there are equations for that, but it's not necessary to show you these equations there. And people have discovered that all these interactions are proportional to this factor, 3 cos square theta minus 1. And it's very interesting. And if you place your sample to annul this term, 3 cos square theta minus 1, this is a 55.7 degree angle. So this is theory, but it works. So if you put your sample not aligned with B0, but at this angle of 55.7 degree, and you spin your sample very rapidly to force all the spin to be at this angle. So this is a technique which has been developed for solid NMR, but we use it for semi-solid NMR, and we recover resolution. So by putting the sample at the magic angle and spinning the sample. So the sample can spin at, for example, we spin at 5,000 Hertz. So this is 5,000 round by second. So this is relatively rapid. So this is like a virtual liquefaction of the sample. The sample is not liquid. So we can see there, we use it for brain samples. This is very interesting. This is an extract of metabolites. So only the liquid extracted from brain. And this is the intact biopsy taken from brain at the spinning at the magic angle. And you can see you have high resolution. This means that the peaks are very sharp, so we can identify all the metabolites which are present. While when you have this kind of spectrum, you cannot identify anything, so it's not useful at all for metabolomics. So we use a lot this method. I will show you an example. This is very interesting for cells, for biopsy, for small organisms also. I have used it for Daphnia. I don't know if you know what are Daphnia. This very small crevette. I don't remember how to say that in English. And we put it into the magnet as a magnetic field at 4 degrees. They sleep because if they do not sleep, they cannot support this the high speed. And we can record some spectra of Daphnia. And we were studying this was an ecotoxicology project. We were studying water pollution of one in Lyon. And these small organisms, which are in water, can give you an indication of the level of pollution in water. So by using metabolomics, so you can see there are a lot of possible applications. So the Daphnia were not, they were alive. After recording the magic angle spinning spectra of Daphnia, we put them in the aquarium, and they develop and so on. So you can do a lot of things with this method. So this is interesting because it is there is no sample preparation. You take your biopsy, you put it directly in the magnet, and sample can be stored after animal analysis. I use it for epilepsy. So I will show you an example. The people who have focal epilepsy have surgery, and the surgeon removes the foci where seizure starts. And I have I have a lot of samples from these patients who had surgery for epilepsy, and we have used this kind of metabolomics with the intact biopsy of the people. And we can do targeted dissection because we use very small 1 milligram or 2 milligram of sample. So this is an interesting method to do metabolomics on intact sample. So this is for semi-solids. This means that a small mobility is needed, a small high rotation. This is not suitable for completely solid sample. This is for semi-solid sample. Like all the biological samples are semi-solid. Most of the biological samples are semi-solid. So we have used that for eyes, for liver, for heart, for skin, for muscle, and so on. So just to show you a magnet. I don't know if you have ever seen a magnet for an hammer. So this is a vertical magnet. Very big because here people would be there. Yes, there. This is a 18 Tesla magnet. This is the probe. This is a magic angle probe. So we put the probe inside the magnet. So this kind of, and there are coils there to exit the spin. And this is a magic angle here where the sample are put. So the sample is inside this small part. So you put the sample in this kind of very small tube. You can see the size of the tube to put the sample. It's very, very small. This is why we can analyze very, very small. Despite the fact that NMR is not sensible, here we can analyze very small sample, and yes, it's quite interesting. So for human tissue, it's very interesting, this kind of, because after that we can do histology or Western blot to analyze proteins after NMR. So we have for Parkinson disease, we use this kind of method. And so we do Western blot after NMR. So we have metabolites and proteins on the same sample. So very robust skins provide very robust results. And the rotors are very zirconium, very hard, because we spin at high speed. This tube is pumped at very high speed into there is a very high speed, so it's necessary that the tube be very, very hard, so it is in zircon. So this is just to show you some equipments, camera equipments, to have an idea of what is you can see this is a big equipment. This is to do liquid NMR. We use this kind of tube. This is for blood there. Yes, this is to see what is the size of a magnet and where the sample is put. With emerald, there is a big hole and people go inside the magnet, but the magnet is horizontal. So this is you can see where the sample go inside the magnet. So this is very, very high technology because the magnet is made, the magnetic field is made with this is supraconductivity. So the coil is in helium, liquid helium, at the absolute zero absolute, to have a permanent field. There is no alimentation, nothing. This is a permanent field. This is the most important application of supraconductivity. This is the magnetic field. And there is also, we must generate frequency, high power frequency to exceed the speed. So this is very, very high technology with different parts. Supraconductivity, electronic, high power electronics, etc., etc. So this is why this is expensive. I would like to make a small stop if you agree with that. So 10 or 15 minutes of stop, and we'll start after that. Okay? 15 minutes with the